with the use of marrow, we replicate and supplement what happens naturally. Well, when we put stem and progenitor cells into a region of trauma, whether it's in heart or it's in bone or it's in cartilage or wherever it may be, the majority of stem cells and progenitor cells and the cells that we've just placed there die over time. And yet, in this study with Turan, who's in Germany, we used bone marrow CD34 and CD133 positive cells. So we had hematopoietic stem and progenitor cells from marrow, put them into the patient. Now look, we have a reduction of infarct size, an increase in the global ejection fraction, increase in infarct wall movement, not just immediately, but 3 months, 12 months, 24 months, 48 months, we now have the data for that's been published. So what we're saying is, even though the cells are not there 4 years later, that heart is still improving. And there's a reason for this. And the autocrine effect is really where people thought of stem cells many, many years ago. And we thought, if we can take a stem cell and put it into someplace in the body, that stem cell will act upon itself and it will grow the new tissue. Okay? That does happen, but at a very, very low percentage, anywhere from 1 to 5% of the cells will act in an autocrine fashion. The overwhelming majority of the regeneration of the tissue that occurs is through what's called a paracrine effect. And that paracrine effect says, when you put these cells into this region, a high concentration of cells actually stimulates quiescent cells in that region to awaken. And they activate, and they are the ones actually responsible for regenerating that tissue.